And in these DNA fragments, finally the bright orange colored bands taken out from this agar gel. So after agar gel making different cuts from each piece of that is agar gel, extraction of the DNA, what do you call it is elution. Now eluted DNA is ready to link with what it is the vector, the carrier molecule to carry to the host cell for expression of it is the foreign gene. So isolation of the DNA fragments, agarogel electrophoresis, ethidium bromide, exposed to ultraviolet rays, bright orange colored bands, cut out the that is agarogel from each piece, extraction of the DNA molecule, what you call elution, eluted DNA ready to be linked with the vector. Next, the vector. The vector is a carrier molecule to carry the foreign gene to host cell. It is a DNA molecule, it carries the foreign DNA. For multiplication of the foreign DNA, what do you call it is the cloning vector. And in case of the cloning vector, we are having bacteriophages as well as the plasmid. The bacteriophages and plasmids which can multiply in the bacterial cells independent of the bacterial chromosome. And if we take bacteriophage having high number per cell equal to copy number of the genome. Suppose the phage or virus having the capacity to multiply in the cell and produce suppose 100 copies. That is nothing but the copy number. So if you link the foreign DNA to this virus, the phage, we are going to get this copies, it is the 100, that is called copy number. And if you take the plasmid having 1 or 2 copies, 10 or 15, even 100, even more than it is 100. So if you link the foreign DNA to plasmid or otherwise the phage, it multiply and produce the copy number of this, what it is the fragments of the DNA, desired DNA, whose number is equal to copy number of the phage as well as plasmid. So thing is, we are linking the foreign gene to phage DNA or plasmid DNA, it multiply and equal to copy number of phage or it is plasmid. Suppose the phage having 100 capacity, it produces 100 copies. The plasmid having 90 capacity is going to produce the 90 copies. So what do you call it is copy number. So vectors are constructed in such a way that we have to identify recombinant DNA from what it is non-recombinant DNA. That is important. And what are the characteristic features of vector? So keep the textbook with you. That is the characteristic features of the vector. The ori, selectable markers, low molecular weight, and the cloning site. First we take the ori, origin of replication. The sequence of the DNA where foreign gene is linked to the ori, that means it initiates the DNA replication. So DNA is not supposed to replicate randomly at any place. It replicates, initiation of replication occurs at a particular place. What do you call it is ori. If you link the foreign DNA to the ori, it produces multiple copies. So ori is responsible for not only the initiation of DNA replication, also responsible for copy number of the linked DNA. Suppose it is a linked, you are linking this DNA, its number decided by it is ori. So ori is responsible for copy number of linked DNA. That is very, very important. So we are going to prefer the vector with ori having high copy number. Next, selectable marker. In case of selectable marker, the selectable markers which are antibiotic resistant genes. Because suppose if you want to ligate this foreign DNA, it is a vector. The vector accepts this foreign DNA. Now it is recombinant vector. And if you want to ligate this vector, it is intact, not accepted any foreign DNA. So some vector accept this foreign DNA, some vector do not accept this foreign DNA. So how many types of the vectors are formed? The vector with insert, recombinant DNA, the vector it is without insert. So, and if it is plated on the medium that is allowed for transformation, some cells are transformed, some cells are not transformed. So, in order to identify the transformed cell, we must have antibiotic resistant gene. The antibiotic resistant gene helps in the selection of the transformant and elimination of non-transformant. So, antibiotic resistant genes, that is the resistance for ampicillin, tetracycline, kanamycin, chloramphenicol. But equally does not have the any antibiotic resistant gene, that is not having the genes for resistance against any of the antibiotics, kanamycin, tetracycline, chloramphenicol, ampicillin, not having any resistant genes for these antibiotics. So it should be restructured. So we had to make this artificial plasmid in order to be ideal. It should have, that is, antibiotic resistance genes. So such plasmid, artificial plasmid, that is PBR 322, plasmid Bolivar Rodriguez 322. So from that laboratory, a discovery number it was 322, plasmid Bolivar Rodriguez. And if you observe the diagram in case of the text PBR 322, plasmid Bolivar Rodriguez, it is a polynucleotide. On the top end, E core 1, CLAV1, Hind3. On right side, tetracycline resistant gene, BOMH1, SOL1. At base, rope, it is having PV2. Beside this, it is ORI. And at left top, it is having ampicillin resistant gene, having PST1 and PVU1. So, E core 1, CLAV1, Hind3, BOMH1, SOL1, PVU2, ORI, and PST1, PVU1. So, how many number of restriction enzyme in case of the PBR 322? 8 restriction enzyme. So, how many cloning site? 8 cloning site. And how many antibiotic resistant gene? Ampicillin, tetracycline. Rope coding for PVU2. And if you want to ligate the segment, you can ligate the segment at any place except at where it is the rope. The rope is a sequence of nitrogen bases coding for the protein useful for replication of the plasmid. So, ligation of any foreign DNA by using PVU2 at a rope site is going to be futile. It is going to be waste. Next, molecular weight. Any vector, it should be with a low molecular weight because it is going to be metabolic burden on the part of the host. Because host always tend to lose what it is the vector, that is the recombinant vector. So it must be with low molecular weight in order to reduce the burden on the host. Next, cloning site. One restriction enzyme preferably have one cloning site. Suppose A is a restriction enzyme. It is having 3 points of what it is in case of the particular vector. So A restriction enzyme at 3 points it is having cloning site mean it is not feasible. It is going to be fragmented. Useful points are eliminated. Suppose A is a restriction enzyme, B restriction enzyme, C restriction enzyme. For A, B, C, for A have one site, B have one site, C have one site, so it is feasible. So one restriction enzyme having one site, it is preferable. If one restriction enzyme more than one site, cloning site, it makes the process highly complex. Some useful points are going to be eliminated and it is going to be fragmented. Next. So, you can link the foreign DNA generally at any one of the antibiotic resistant gene. So, we are having ampicillin as well as tetracycline. Suppose you ligated by using BOMH1, it act at tetracycline resistant gene. So, by using BOMH1, you are ligating this foreign DNA at a tetracycline resistant gene using restriction enzyme BOMH1. So, it is going to lose tetracycline resistant gene as inserted at a tetracycline resistant gene. So, it is going to have ampicillin resistant gene. So still it can be selected out by using ampicillin resistant gene. So you inserted the gene at tetracycle and is going to lose one tetracycle resistant gene. Still it can be selected out from it is by using ampicillin resistant gene. So whatever the cells first growing in ampicillin medium transferred to tetracycline. So recombinants as well as non-recombinants grows in case of ampicillin. Non-recombinants grows in both ampicillin and tetracycline. But recombinants grows only in ampicillin but do not grows in case of tetracycline. So in this case, ampicillin resistant gene is a selectable marker. The gene which got inactivated due to insertion of the foreign gene for selection of recombinants. So one antibiotic resistant gene helps in the selection of transformant. Other gene which got inactivated due to insertion of the foreign gene helps in the selection of recombinant. But selection of recombinant by this process, the insertion inactivation of antibiotic resistant gene, simultaneous plating on both antibiotics is a cumbersome process, that is unmanageable process, because simultaneous plating it is required. So non-transformants would not grow in any medium. Transformants and recombinants grows in case of ampicillin but do not grows in case of tetracycline. As non-recombinants grows in both ampicillin as well as tetracycline, because ampicillin resistant gene present, tetracycline resistant gene present, grows in both ampicillin as well as tetracycline. You will see this in case of the diagrammatically. You see in case of the first diagram A, second diagram B and C. In case of A and B, both are transformants and C it is non-transformant. In case of A, we can expect recombinants as well as non-recombinants. In case of B, we can expect non-recombinants but not it is recombinants, because tetracycline resistant gene which got inactivated. And non-transformed cells will not survive in case of the C. So one antibiotic resistant gene helps in the selection of transformants, it is ampicillin resistant gene. The other gene which got inactivated due to insertion of the foreign gene at tetracycline resistant gene that helps in the selection of what it is recombinant. So A, B, C indicate what? A, recombinants as well as non-recombinants in case of A. In case of the B, non-recombinants grows only in case of, or it is the B, but not recombinants. C, that is non-transformant will not survive in case of medium, it is the C. So as it is a cumbersome process, some alternate selectable markers are discovered, that is ability to produce color in presence of chromogenic substance. What is chromogenic substance? X-gall. X-gall is hydrolyzed by the enzyme, it is beta-galactosidase. The beta-galactosidase, the enzyme is coded by LAC-Z gene. So LAC-Z gene code for beta-galactosidase. Beta-galactosidase is used in case of the medium. It hydrolyzes X-gall into blue color. Suppose it is the plasmid, it is the foreign gene, and it is the LAC-Z gene. So in this LAC-Z gene, it is the LAC-Z gene. And in this, we inserted the foreign gene. So it lost what it is LAC-Z gene. In another case, the vector do not accept any foreign gene. LAC gene it is intact. So as it is having LAC-Z gene, the transcript is formed, mRNA is formed, beta-galactosidase is formed. As it is not accepted any foreign DNA, so LAC-Z gene is going to be intact. So in one vector, as it is an insert, it is without LAC-Z gene. Suppose if we take the one experiment, in this the plasmid, it is LAC-Z gene. In this we deleted this LAC-Z gene in this foreign gene it is inserted. So there is no transcript for lag Z gene, and there is no translation product, that is beta galactosidase. So whatever the colonies in case of the cells which are colorless, white, what do you call it is recombinant colony. Because the plasmid, it was not intact, it was interrupted by the foreign gene. It is without lag Z gene. So the colony is going to be recombinant colony, that is colorless or white colony is going to be the recombinant colony. Next, as it is going to the lag Z gene, it is going to have transcript, translation produce beta galactosidase, hydrolyze X-gall into blue color. So non-recombinant colony is blue color, recombinant colony is white color. In recombinant colony means the plasmid which got disrupted by foreign gene. In non-recombinant colony, the plasmid having intact lag Z gene. So white or colorless colony is recombinant and blue colored colony it is non-recombinant. You see the diagrammatic representation. You see the diagrammatic representation of it is insertion and inactivation of beta galactosidase. In one vector there is an insert, that is foreign gene. In another vector there is no insert, lag Z gene it is intact. So recombinant colony is going to be white or colorless, non-recombinant colony is going to be blue color. So as in case of the recombinant colony it is white color and the non-recombinant colony it is the blue color, because X-gall it has to be hydrolyzed by beta galactosidase. The beta galactosidase is coded by lag Z gene. So it is alternative method for identification of recombinants from non-recombinant based on the color. Okay, non-recombinant blue, recombinant it is white. In non-recombinant the plasmid it is intact, that is having lag Z gene. In recombinant, lag Z gene it got disrupted as it is inserted with the foreign gene. So these alternate selectable marker helps in the selection of recombinants from non-recombinant based on color. Next, in next heading, the vectors in plants and animal. As you are familiar that the viruses and bacteria, it transfer the genes into host cell, that is eukaryotic cell, and direct these eukaryotic cell to synthesize the chemicals as required by this pathogen, that is the virus and that is the bacteria. If you know the art of the delivery of the genes into the host cell, certainly it is going to generate the knowledge which tools are responsible. We can modify the tools to make it a suitable vector to deliver the gene of interest into plants and animals. Suppose if we take the example, Agrobacterium tumefaciens, it is a natural genetic engineer. TI plasmid, tumor-inducing plasmid. In that particular DNA fragment, that is tDNA, it is the plasmid, TI plasmid, it is tDNA. It is the tumor-inducing plasmid, TI plasmid, it is tDNA. So it is the tool, it is the tDNA. The tDNA is going to be responsible for converting normal cells into tumor cells in most of the dicot. So Agrobacterium tumefaciens is a pathogen for several dicot plants. So we are modifying the tools. So in this, we are deleting what it is, the gene, that is tDNA. So it is the TI plasmid, the tool. It is the tDNA. So in this, so it is the TI plasmid, the tDNA. So this is responsible for conversion of normal cells into tumor cell. As the tDNA got deleted from the TI plasmid, in the place of the tDNA, the foreign gene of interest it is inserted. So we can deliver the gene of interest into the variety of the plant cell. As this vector it made disarmed. Earlier it is a pathogenic vector. Now it is disarmed. It is a tool got modified. Which tool got modified? Ti plasmid got modified. Which segment got deleted? tDNA got deleted. In that place, what inserted? Foreign gene it is inserted. So tool got modified to deliver gene of interest into variety of the plant, dicots as well as the monocot. The key points are: the Agrobacterium tumefaciens is a pathogen for several dicots. It is a harmed vector. Now it made disarmed by deleting what? That is tDNA. You modified which tool? Ti plasmid. So it is used to deliver the gene of interest into variety of plants. Next, retrovirus. It converts normal cells into cancer cells. Whatever the gene responsible for converting normal cells into cancer cells, it got deleted. In that place, particular gene it is inserted. So disarmed vector. So retrovirus, it made disarmed to deliver gene of interest into animal cells. The 2 point. Delivery of genes into plants, Agrobacterium tumefaciens, Ti plasmid, tDNA got deleted. Retrovirus, it also made dissolved by the deleting of the gene responsible for it is, that is the cancer. Next. So the plants and it is animals. And next heading, competent host. That means the host must be made capable to accept it is the foreign DNA. Suppose it is a bacterial cell, it is the foreign DNA. The DNA is hydrophilic, charged. It cannot pass through the membrane of the bacteria, because the membrane of the bacteria it is having what? Hydrophobic tails. A DNA is hydrophilic. It cannot pass through the hydrophobic tail. So the cell it must be made competent to accept this DNA. Making the cell competent to accept this foreign DNA, what do you call it? Is competent. How can you make the cell so efficient to accept the DNA? By treating with specific divalent calcium ion. Specific divalent calcium ion, that is calcium chloride solution. So what are formed? Transient force are formed in case of bacterial cell wall. And how can you make this DNA forcefully to enter into the cell? By plating or incubating on ice, heat shock 42 degrees centigrade, then ice, and the bacterial cells can accept what it is the foreign DNA. So we are increasing the efficiency of the cell by treating with specific divalent calcium ion. Then it accepts what it is the foreign DNA. The cells are said to be transformed. So specific divalent calcium ion, it makes transient pores on cell wall. DNA forced to enter into the cell by incubating on ice, heat shock, ice. In heat shock 42 degrees centigrade. So this method is employed in bacterial cells. Next, in case of animal cell, rDNA directly injected into the nucleus of animal cells. That is called micro-injection. Next, in case of the plant cells, the method it is biolistic shotgun gene gun method. In this we are going to use high velocity particles, microparticles like tungsten and gold. The tungsten and gold are coated with DNA. So DNA coat around tungsten and gold. And by using the gun, gene gun, the plant cells are bombarded with the tungsten and gold coated with what? DNA. So plant cell can accept what it is or DNA. So 3 methods in bacteria: ice, heat shock, ice. In earlier, calcium, specific calcium ion treatment. And second, micro injection refers to animals. Biolistic gene gun method refers to plant. At last, disarmed vector method. Earlier we said Agrobacterium tumefaciens, it made disarmed, Ti plasmid got modified, tDNA got deleted to deliver gene of interest into plant cell. Other disarmed vector it is retrovirus to deliver gene of interest into animal cells after making disarmed. Next. In next heading, insertion of recombinant vector into host cell. So after making the host cell competent by treating with different methods, that is calcium treatment, ice heat shock, ice method, and different methods. So after making the cells competent to accept what it is the foreign DNA, and suppose the recombinant DNA bearing antibiotic resistant, it is a recombinant DNA foreign gene having antibiotic resistant gene inserted into the bacterial cells. After making the host cell competent and introducing recombinant vector into the bacterial cell by different method, the cells having recombinant vectors bearing antibiotic resistant gene. So it should be plated on agar plate having antibiotics. Only ampicillin resistant gene, the cells having ampicillin resistant gene only will be survived, and others that is not having antibiotic resistant gene would not survive. So in this context, ampicillin resistant gene is considered to be a selectable marker. Why we must have these antibiotics in case of medium? Because the bacterial cell, it is with a recombinant vector, the foreign DNA. The cells always tend to lose the recombinant vector because providing metabolic, it is a metabolic burden on the part of the host. So always tend to lose it. In order to maintain, in order to retain the recombinant vector in case of the bacterial cell, the medium it must be poured with the antibiotic. So in order to resist this, it must have antibiotic resistant gene. It is without antibiotic resistant gene, the host tend to lose what it is the plasmid. So in order to maintain the recombinant vector in host cell, it must have antibiotic resistant gene in vector, and in case of the medium it must have antibiotics like it is ampicillin or tetracycline. So in this context, so what do you call it is ampicillin resistant gene is a selectable marker. So in this paragraph, the few key points which are the ampicillin resistant gene is a selectable marker. And before that we are having polymerase chain reaction and how we are going to isolate this DNA. Actually in case of the process, we have to isolate the DNA, fragment the DNA, and isolation of the desired DNA fragment, insertion into vector, insertion of recombinant vector into host cell, and plating them on that is a culture medium in order to get the product. So undoubtedly nucleic acids are the genetic material in all organisms without any exception. DNA is the genetic material in all organisms. That is going to be wrong. DNA is a genetic material in most of the organism. And DNA is only the genetic material in all cellular organisms. This is going to be the right. So in most of the organism, DNA is the genetic material. In all cellular organisms, DNA is only the genetic material. So first we have to isolate the DNA from the cell. Where DNA is located in the chromosome, where it is present in the nucleus, surrounded by nuclear membrane, in turn surrounded by cytoplasm, which in turn surrounded by membrane, in turn surrounded by cell wall. So cell breaks open to release the DNA in a purified form. So why you are going to opt this the purified DNA only? Because restriction enzyme it can act on the purified DNA but not on RNA or proteins or some other lipids or carbohydrate. So cell breaks open to release the DNA in a purified form. So cell it should be digested with cellulase in case of plants, chitinase in case of fungi, lysozyme in case of prokaryote. So in case of eukaryote, plants and fungi, cellulase and chitinase. And in case of prokaryote, it is lysozyme. Next, plasma membrane, detergent lysis of the plasma membrane. And by using protease to digest the protein, RNase to digest RNA, lipase to digest the lipid, carbohydrates to digest the carbohydrate, and but not it is the DNA ases, because we are going to have the DNA, but not we're not going to use the DNA ase. So RNase, protease, lipase, carbohydrates, but not DNA ase. And subjected to gradient centrifugation in a chilled ethanol, purified DNA precipitated out, and the purified fine threaded DNA taken out from the test tube in from suspension by using a glass rod by stirring motion technique. As DNA is so sticky, by stirring motion technique to the glass rod, the fine threaded DNA forms a spherical ball around the glass rod. What do you call it? Is spooling. So in chilled ethanol, purified DNA precipitated out. By using glass rod by stirring motion technique, the purified DNA, that is fine threaded DNA, forms a spherical ball. What do you call it? Is spooling. So after spooling, it is ready for fragmentation. So host DNA and vector DNA cut with the same restriction enzyme in space, that means differently, distantly, combined, that is mixed, and added with the DNA ligase, what are formed? Recombinant DNA molecules are formed. Suppose if you use the restriction enzyme A for host, B for vector, undoubtedly in case of the host and vector, sticky ends are formed, but the sticky ends are not matched, unmatched. So even though if you use the different restriction enzyme in host and vector, sticky ends are formed, but it is unmatched, not going to be complementary. If you use the same restriction enzyme A in case of host, A in case of vector, sticky ends are found which are ligated, readily undergo complementation. So unless you use the same restriction enzyme in host and vector, recombinant DNA molecule is not going to be formed. If host DNA and vector DNA needs to be amplified to several billion copies, we are going to opt the method. It is PCR, polymerase chain reaction. It was developed by Carey Mullis. In that requirement, it is thermocycler, a computerized machine, deoxynucleotide, DNA polymerase. In this, we are going to take a tech polymerase. It is obtained from thermostable bacteria, that is Thermus aquaticus. It is a eubacterium. It survives in case of varieties hot springs. The tech polymerase is active even during high temperature denaturation of DNA. Tech polymerase is active only high temperature, that is going to be wrong. Tech polymerase is active even during high temperature denaturation of DNA obtained from Thermus aquaticus. It is a eubacterium, it is not a archaebacterium. That is one point. And we require two sets of the primer. The two sets of the primer which are deoxynucleotides, not it is RNA primer. In earlier DNA replication, we used this in case that is in vivo, that is RNA primer. It is in vitro, it is the DNA primer, oligonucleotides. So, oligonucleotides which are the DNA primers. Next. And the steps which are denaturation, annealing, extension. The genomic DNA itself acts as a template. And in denaturation, hydrogen bonds are broken, two strands got separated. And whatever the primers, deoxynucleotide, a oligonucleotide, it is going to attach it to third end of DNA. Both primer, two sets of the one set of the primer to third end, other set of the primer also to third end of the DNA. What do you call annealing? Otherwise, what do you call hybridization? So, denaturation and annealing or hybridization. And third step, extension. Whatever the enzymes used, tech polymerase, whatever the deoxynucleotides available in case of the medium, the tech polymerase extends primer at 3OH by adding what? Deoxynucleotide. So, primers are attached to DNA at third end. Primers are extended by tech polymerase. Deoxynucleotides are added to third end. So, this DNA formation in case of 5-3 direction, addition at what it is the third end. So DNA it is amplified to several billion copies. After 30 cycles, you are going to get nearly 1 billion copies of the DNA. So, if any vector or host DNA needs to be amplified, the method it is PCR, polymerase chain reaction. It is not photosynthetic carbon reduction, it is polymerase chain reaction, denaturation, annealing, extension. Tech polymerase withstand even during high temperature denaturation of DNA. Next. And as you inserted this recombinant vector into host cell, finally we are going to get the gene product. And if you inserted the foreign gene into the vector, transfer it to host cell, it will multiply. We inserted the foreign gene in vector, this recombinant vector transferred to the host cell, it will multiply. So ultimate aim in case of recombinant DNA technology is to produce the gene product, that is nothing but protein. So having optimized the condition, because recombinant DNA needs some technical steps in order to express, we provided all the technical steps, and now we are thinking to produce varieties in large scale, the large biomass, after having optimized the varieties the condition. Whatever the gene encoding for particular protein expressed in heterologous host, what do you call it is recombinant protein. That is also a key point. The gene encoding protein expressed in heterologous host, what do you call it is recombinant protein. That is one point. And in case of the small scale, in case of laboratory, the cells having recombinant vector, the cultures are used to extract the material. By using different process, we are going to extract this. It is a small scale, applicable in case of the small scale, that is laboratory condition. Cells can also be multiplied by continuous culture system. The word itself may conveying its meaning. That is, from one side the user medium it is drained out, in other side fresh medium it is introduced. So it keeps the cells physiologically active, exponential, log stage to produce larger biomass. So high biotechnological product you are going to get. So user medium drained out, fresh medium introduced. What do you call continuous culture system? To keep the cells in physiologically active, exponential, log stage for high biotechnological product for larger biomass. Next. So small volume culture does not need appreciable volumes. So for sake of large volume culture, that is 100 to 1000 liters, which are processed in a container, that is the vessels, what do you call bioreactors. So bioreactors is thought of, it is the vessels, stainless steel vessel, and in this raw materials are biologically converted into products by using plant cells, microbial cells, animal cells or bacterial cells. You are going to get the product like vaccine, enzymes or proteins. So raw materials are biologically converted into products in bioreactor. So we have to create congenial conditions in case of the bioreactor, temperature, pH, salts, nutrient, aeration, everything is going to be provided in case of bioreactor. And there are the two types of the bioreactor, stirred tank bioreactor and sparged stirred tank bioreactor. You see in case of the diagram, in case of the text, the stirred tank bioreactor, cylindrical and curved at the base, help in mixing up of the contents present in the container. That is one point. And the stirrer, that is, it is having a knob, that is a motor, and a rod, stirrer, and a blades. Collectively, to what do you call it is an agitating system. So the stirrer facilitates even mixing, oxygen availability throughout the container. That is another point. So it is having agitator system. That is the motor, the rod, having what it is the different blades, what do you call agitator system. So it is an agitator system, foam breaking system. If you observe in case of the diagram, upper blades, the 4 blades, which are meant to break what it is the foam. In case of the lower blade, what do you call flat blade impeller, as it is going to have the 6 blade, 2 long blades and 4 short blade. and it helps in the circulation of the broth. So upper blade meant for foam breaker, lower blade meant for circulation of the culture broth. And alternatively, air is pumped into the container, sterile air is pumped into the container. Acid base control system, that is pH control system, oxygen delivery system, foam breaking system, temperature controlling system, everything which is available in case of the stirred tank bioreactor. So there are some ports, the places, inlets and outlets, because as it is a large container vessel, every time we are not going to take all the contents. So for periodical testing, we have to take the what samples out from the container. So there must be some inlets and outlets. It is a continuous culture system. That's why it is having inlets and outlets. So samples are collected periodically for testing from the places or the ports. What do you call it is sampling ports. So in case of stirred tank bioreactor. In second diagram, we observe sparged stirred tank bioreactor. Sparging by forcing of air, we are creating increased surface area for oxygen. So oxygen transfer rate is increased and gas entrainment will occur. 1, 2, 3 points in case of sparged stirred tank bioreactor. So stirred tank bioreactor, sparged stirred tank bioreactor. Labeling it is very, very important. Upper blade, lower blade, acid base, steam for sterilization and sterile air is sparged stirred tank bioreactor. 3 points. Okay, by forcing of the air, bubbles are created. Bubbles dramatically increase the oxygen transfer, oxygen surface area. So oxygen transfer area will increase, then oxygen, the gas will be entrained. And at last, downstream processing. Before released as a finished product for marketing, first upstream process up to biotechnological product obtained. This is upstream process. And whatever the steps in between, that is biotechnological product and marketing, what do you call extraction and purification, collectively what do you call downstream processing. Extraction and purification. Next, it should be formulated. The ingredients must be inactive, physiologically active. And these ingredients, so as to be active, it should be formulated by in case of preservatives. So proper formulation in case of by using preservatives. Next, in case of the drugs, clinical trials in case of the drugs, and strict quality control testing for any biotechnological product. That is one thing. At last, downstream processing varies from product to product. So in this total topic, you are going to have some key points in every paragraph wise. So try to memorize, try to feed the information as the frames, different frame. First paragraph, what is the gist? Second paragraph, what is the gist? And third, fourth paragraph, like this. So I hope you feel comfortable. Certainly you are going to recollect this information very easily. So hope to see in case of the next videos. So subscribe and share. All the best.